So I was an undergraduate at Stanford University, and I was just there right before Colin Pittendrigh actually came. The year that I was leaving, I heard a rumor that this wonderful biologist had been stolen away from Princeton and was coming to Stanford. And in a course I was taking on comparative psychology, the professor said, I'm uncomfortable even talking about biological timing because the world's expert will soon be here. Nonetheless, I will tell you about biological timing. And he gave a lecture on biological timing, which I thought was very interesting. However, I was in psychology, and I was interested in experimental psychology. And I went to the University of Oregon to actually work with a perceptual psychologist to study three-dimensional space and how it's represented on the retina and how one interprets three-dimensional space. And I began doing experiments in experimental psychology, and within 3 weeks I realized I had made the mistake of my life. I would never want to be an experimental psychologist, and the work was boring, and the models were hard to validate, and I began to desperately look around for a new laboratory to work in. So I wandered into a lab in a literally an aluminum hut, actually, on the other side of the mill race, which was a river that divided campus. And there was Marvin Lickey, who was a Felix Stromwasser postdoc. And Lickey was recording from individual neurons in a marine snail called a neuron called R15 in the abdominal ganglia that was a circadian clock. And I looked at the equipment, and I listened to Lickey a little while, and I decided I had to do this. This is really what I wanted to do. And I think I had been primed by the lectures I had received at Stanford, and I remember hearing about Colin Pittendrigh. And Lickey and I became quick friends, as well as he was my mentor. And I stayed in his lab actually for 5 years and got my master's and PhD. And it took a little bit of a change in direction. I mean, really had to learn. I had taken a lot of biology courses as an undergraduate, so it wasn't so challenging, but this was a really different area for me. And I'd say I was hooked immediately. I mean, as soon as I started looking at the possibility of recording from individual cells, and I liked the technology, I liked the questions, I thought it was really a great area. And Lickey was a good mentor, actually, and not well connected into the circadian rhythms world, but actually a very good mentor. When I graduated, about a year before I graduated, I had to decide what I was going to do with my life. At that point, I decided, of course, I'm going to do a postdoc. And I applied to about 4 postdocs. I think I applied to Irv Zucker's lab, and that was going to be more traditional circadian rhythms. I applied to Zucker's lab. I applied to Stromwasser's lab, actually at Caltech, thinking about going there. Then I applied to Colin Pittendrigh's lab, but also Don Kennedy's lab, because I was also interested in neurobiology, and Don Kennedy, I had him as an undergraduate. He was a great neurobiologist, and I thought I might be interested in actually doing neurobiology, maybe trying to combine it in some way with circadian rhythms. So Zucker was the first person to come back and give me an offer, and it was very attractive, but it was pretty traditional in terms of what I'd be doing actually at Berkeley. And then Pittendrigh didn't respond. I don't think Pittendrigh actually responded, but Kennedy responded. And Kennedy said, we've got a group we're putting together. Terry Page is coming here, and that I'm trying to put together a group between Pittendrigh and myself. We're going to have a small group of people that will do both neurobiology and biological timing. And I jumped at it. So what an opportunity to work in both labs. So moved down to Stanford, and I really began working in Don Kennedy's lab and working on crayfish and working more on locomotor systems than on circadian rhythms. Pittendrigh was half interested in me. He was busy, he was writing, he was just finishing up papers. I think that was the Sergey Don and Pittendrigh papers. He was still going through page proofs on them, so didn't show much interest in my activities. But little by little, Pittendrigh and I got to know one another. And Kennedy eventually left for the FDA. He became FDA Commissioner of the Food and Drug Administration. And he left, and I moved to Pittendrigh's lab, so full-time. And then Pittendrigh accepted the directorship at the Marine Station, Hopkins Marine Station. And Page and I think, were the main people that moved down with Colin at the time. So the 3 of us sort of moved the lab, set up shop. Carl Johnson came, I think, the next year.
What a trio.
Yeah, I think Carl Johnson. So Carl Johnson came. And I think Carl might have been actually a first-year graduate student when I was up on the main campus. But he actually moved with us to Monterey, or he came slightly after we did to Monterey. And Carl was great. So there was Carl, there was Terry, there was myself. And I think Jeff Elliott might have come with about another year delay. So Elliott was there. So I think there's the 4 of us there all at one time.
And this was when?
This was probably 1976, something like that. So we were all at Hopkins Marine Station, and Pittendrigh was just setting up to have a big launch for the Marine Station, and he thought the best way is to have a course on biological timing, and he organized this summer course in 1977, which occupied all of us for a full half a year. You know, rather than doing research, we were essentially getting ready for the course, which was an enormous success, but we put a lot of work into it, and it was a great time. And Pittendrig's lab was incredibly unusual in the sense that, unlike a lot of postdoc labs where you actually work on a project that your postdoctoral advisor is working on, Pittendrig's attitude was very different. When he essentially told me, "Chum, work on whatever you want, just don't work on my problem." You know, he actually chum.
That's
work
the word.
work on your own problem. And he was really, in a sense, it's wonderful when you think back. I mean, Colin was interested actually in you being there for sort of intellectual stimulation, for his intellectual stimulation, for your intellectual stimulation. You were not a cog in a wheel. You're not a lab person who was providing data for his projects.
Or slaves or any of that.
Unbelievable. Yeah, the difference has changed so dramatically. I mean, there was no conflict between mentor and employer. In other words, Colin, you know, nowadays we have the problems in labs where you employ people on your grant, and their careers and your careers are mixed up in unusual ways, and you have to be always very careful whether you're really acting in their best interest as opposed to your best interest, because you often people in your lab are working on projects that you have such a direct stake in. And this was so different. You were there mainly for intellectual purposes, and Pitt was an unbelievably good mentor in that sense, because he disconnected your interests and his interests, and he was just there really to help. One of the great advantages in the 70s was this grant support wasn't so much of a problem. So people didn't have to be working on your grant. Now, I mean, so it really was a different era, and we can think back and say it was wonderful, because mentors were mentors. They weren't employers. You weren't working on Pittendrig's grant. We had our own support. And it was a remarkable experience. And it was every day at 2 o'clock, Pitt would sort of walk out of his office, come in. I think Terry Page and I shared an office, and he'd always start off the same. He'd say, "Chum," he'd say, "Tell me what you're doing." And you'd start talking about your experiments. You'd talk for about 3 minutes, and Pitt would say, "Well, let me tell you what I'm doing." And for the next 2 hours, you'd listen to the Drosophila experiments, and you get an education. I mean, a remarkable education. And Pitt would mix this all up with some of his complaints about what was going on at the Marine Station. He was the director at the time. And you get some complaints about people in the field that were bothering him. And, a tremendously emotional person. And it was fun. I mean, he was very human, and he certainly had lots of frailties, but enjoyable to work with. And I thought those were 3 years that were just the most valuable years, and certainly my time in science. And you realized you were working with a master. I mean, it was pretty obvious you were working with one of the world's great biologists. And he was amazingly insightful. He rarely made a mistake. I mean, his insight was just unbelievable biologically, just amazing. And his intuition was so good. It was always interesting. Don Kennedy, the comparisons were very interesting. And Don is an unbelievably bright individual who's totally plugged in. So you can walk into his lab after not having been there, not having seen him in 3 or 4 weeks, and he'll say, "Gene, the last time we met," I know, he said, last time we met, you were talking about A6 interneuron in the tail of the crayfish, and I just want to know how those experiments went. And I used to think, how could anyone remember all that detail with 20 people in his lab? So unbelievably powerful sort of online processor. But biological intuition, he's right sometimes, he was wrong sometimes. Pittendrig could barely remember your name sometimes, but when it came around to intuition, he was remarkable. I mean, he just understood biology. And, you were in the midst of a genius when you were around him. And that said, I mean, he could be very taxing because he was very emotional. And he had health problems off and on. So, there was always issues we were worried about him. But it was a real gift to be in his lab. So I was there for 3 years. After 3 years, I was running out of my postdoc fellowship, which was 3 years in duration. So I had to look for a job. So started applying for jobs, and the first job I saw was a job at the University of Virginia in Charlottesville, which I had never been to. In fact, I had barely been to Virginia. Virginia was the South in the U.S., kind of an unusual place to go, even in the 1970s. And I said to Pittendrig, I said, "Tell me about the University of Virginia." And again, he says, "Chum." He says, "It's really a pretty place." He says, "I don't know much about the biology department, but I suspect it's pretty good." And I said, "You should definitely apply." So I applied, I got an interview. I liked what I saw, and I went there. So I actually arrived, I think, in Virginia in 1977, and I've been there ever since.
You've been there
ever since. So.
And you had an influence. They were courses that people came from all over,
That's right.
and a whole generation of biological rhythm researchers were labeled.
That's right. It almost defined your
It
position in the
defined the
field.
field.
Were you there or were you not there? Either you were there, or usually if you were not there, it was usually people who were younger who, still weren't at the point. And certainly, you weren't there. There wasn't everybody there, but there was a lot of people came through. Not so many Europeans, some Europeans, not. It was really mostly an American course at that time. And that's how the field has changed so dramatically. If you organized a course today, it would have such different composition. There'd be huge investment from Japan. Europe would have a huge number of people. It really was more insular in those days. I think we had separate communities that were working in parallel but not integrated in the same way.
Well, Aschoff and Pittendrigh, the great dialogue of the Blood Brothers who argued very loudly,
Yep.
They represented the American and the European community.
That's right.
And
But they weren't brought together so well as they are now, do you think?
it's entirely different now.
Right.
When you think then, people didn't travel to meetings quite
That's right.
promiscuously
That's right.
as now.
Yeah. It's interesting. Probably part of it is travel money was more restricted. It was harder. It's just easier. Fares have come down. Cost of travel has gone down proportionally over the years. That's true. It was unusual to travel the international meetings, certainly for students.
And it was a bit of a one-way traffic. The Europeans would come to America.
That's right. That's right.
And not that many went to Aschoff's land.
That's right. Some did, but it wasn't as large a.
Yes.
This wasn't as large a transition as now, where you see much more equal exchange. And Japan was still really beginning in circadian rhythms in those days. There was a few labs, but there's been a huge explosion in Japanese labs. And as you were hearing at the conference, there's a remarkable science going on that is new.
A lot of it's been supported by the Honma.
That's right.
Sapporo Symposium.
Yeah, they were influential. The Hanmas were core in actually changing, I think, Japanese circadian sort of science. And I wonder if everyone appreciates the contribution they've made, which is just enormous when you look at it. And a lot of it very selfless. They've set up a lot of labs. A lot of people have benefited from their efforts all over Japan. It was quite remarkable. Returning to me, so so the courses were wonderful, though. The summer courses were, I think, transformational for many of us, in addition to a lot of fun. And I did not return to them. After I went to Virginia, I don't think I ever returned to a summer course. I think there might have been one more.
The original giver of a summer
We're the original 1977, the special course that everyone remembers so well.
And all the rest were your children, as it were, one
That's
or
right.
2 years
That's
younger.
right. That's right. In fact, we had a course.
Who was there? If you make a list,
Who is it?
it would be all the people who are now running their own business.
Oh, yeah, so the original 77 course, if I remember, and it's hard to remember now. There was obviously Benji Rusak was sort of who was a postdoc in Irv Zucker's lab, I think. Might have just gotten his own position, but he was pretty junior. And Jay Dunlap was a postdoc in Woody Hastings' lab, and he was there, along with about 3 other of Woody's postdocs. Jerry Page, of course, was there. Jerry Feldman was there, who's now, I guess, retired. Konopka was there. Ron Konopka, I think he is a new postdoc at the time, a new a new faculty member, I guess, at Caltech at the time. On and on. It was just one after another. And the number of people that were successful from that group is quite remarkable. You look at the number of labs that were set up. It created a family. Sergey was there, I think, the whole time, because Sergey had spent, of course, several years at Pitt's lab. Before that, he was gone about a year or 2, and then came back to the summer course. He was there. Andreas Aschoff was there. Jorgen's son spent the year a year or 2 years with us, I think, at the Marine State. He was sent by Jorgen at some point when I think his studies were flagging a little bit. And he was a remarkable character. We had a lot of fun. I think he arrived and disappeared the first week to find an excellent beer, and then arrived to find the best U.S. wine. And then very enjoyable working with him, actually. And we all grew close to one another. But then I moved to Virginia, and then it was insular for a while. You're setting up your own laboratory, and worked hard to do that. Basically stayed in the lab for about 5 years, just getting started.
Bulla work.
That's right, and that's why I discovered Bulla and began collaborating with neurobiologists, actually, in the biology department. But after about 10 years there, I decided we really needed to grow biological timing, because I was successful, but I was sort of alone. And sort of with close agreement of several of our other colleagues in the department, we talked about hiring a chronobiologist possibly to lead our department. In other words, bring a person in who's very senior, who could lead the department, but help build chronobiology at Virginia. And of course, that person was Mike Menaker. So we all had a big discussion about whether we could get Mike to come to Virginia. And eventually, with some persuasion and a very good offer, he did. He came. He and Shirley came, and both played an important role, because Shirley immediately moved into the administration. It was actually in the office that I'm in now as provost. He was in the provost's office. It really had enormous influence on the institution. And Mike came, and Mike began to build the group. And it was quite exciting. I think he came about 1988, something like that, late in the 80s.
You are the one who's been responsible,
Well,
sir.
a lot of people were responsible, but I helped put it all together. I mean, really, it was everybody working. About 1991, we started talking about creating an institute. And the first institute we formed was something called the Biodynamics Institute, which is a funny name, which was recommended by Ernst Nobel, who was a consultant, said, have a name that can be anything, because that'll give you flexibility on how you grow. And we had endocrinologists involved and circadian rhythms people. It was sort of a very diverse group of sort of people interested in rhythmicity, from endocrine rhythms to neural rhythms to circadian rhythms. That institute was a good start. The university invested about $2 million in the institute. That gave us actually a basis for hiring postdocs. And when the NSF Centers programs became available, we were in a good position to apply. So we applied for a center, and this was first going to be a UVA center, University of Virginia center exclusively. And it didn't get funded the first round because they said there wasn't enough molecular biology. And in those days, we're thinking, why does it have to be so much molecular biology? You know, most of what's going on now is hypothalamic work and electrophysiology and anatomy and behavior, and the molecular business was still pretty small. But NSF thought it was weak in that area. So the next time we applied, we brought in Mike Young and Joe Takahashi and Fred Turek, and we thought that would give us strength that would satisfy the reviewers, and which it did. And that was an amazing group, the original group. Mike Young turned out to be heroic. I mean, he was one of the most remarkably generous people with his time, helped us a great deal. Takahashi delivered beautiful, beautiful material for us for the grant, and Fred, of course, works unbelievably hard. So we had three of these superstars coming in, all willing to work hard, and it came together. And I think we launched in 1993. We thought it was an act of God when we were funded. These are very competitive. There were only 25 of these in the entire country. And to get one in biological timing really was just unbelievable. And I remember the day we found out about it, it was unreal, because it was a lot of money in those days, and it was guaranteed support for 11 years if you could make good progress. So
it defined the field, it brought it out into the establishment. The timing was right for the timing.
That's right. That's right. It was, and it made a huge difference. But it didn't come easily. And the focus of the center, which I think was eventually very important, identifying clock genes, didn't come naturally. You know, at the very beginning, we were very immature, scientifically immature, and really administratively immature. And we ended up with a program that was a little bit for everybody. We were really the first year dividing the money N ways. And everybody was going off in their own lab and doing their own thing, just like a lot of centers that are not going to be successful. The first year site visit from NSF, which was the worst experience of my life in many ways, because they were so critical of what we were doing. I mean, we had thought we'd made progress, because we had some big finds our first year. You know, we'd shown that single neurons in bulla, for example, were competent circadian pacemakers. There was a lot of interesting things going on, but none of it was really cooperative and coherent. We weren't operating like a center. We were operating like little fiefdoms, with everybody having their own little bit amount of money to work on. And they essentially told us, NSF, get your act together, or you're not going to continue. And they told a lot of centers that. We weren't the only one. And we panicked, and we brought in our advisory board, and our advisory board had great people. Bob Moore was on the advisory board. Marty Zatz was on the advisory board. Jay Dunlap was on the advisory board. And they were tough. But they sat down with us in about a 3-day meeting and planned the Clock Genome Project. So, you know, I will not take credit for that. I don't think any one person takes credit for that. I think that was the advisory board and the scientists sitting down and working together. And the amount of time that, for example, Jay Dunlap spent is really enormous, actually, to help us. So this advisory board, which was getting nothing for this other than a trip to Charlottesville and, you know, a lot of aggravation getting there, was really fundamental, actually, in getting the center on the right track. And once we got on the right track, things began to happen very quickly. You know, we hired Steve Kay, and that was one of these hires where when I got done with that, I thought, I can't believe I've just done this. You know, we brought in one of the really brightest young people around. He and Shelley Halpane, his spouse, both enormously good hires, and that made a huge difference because Steve then began to make connections with other people that created technology and interesting applications. So we moved very quickly from there. But all I had to do was sort of keep all these, you know, strong personalities apart, keep them working together without on one another's throats. And a lot of this was just being a referee And it was challenging. Some of the meetings were remarkably challenging because, especially later on when the group got larger, you know, we included Brandeis later on. Scripps, when Steve moved to Scripps, we included Brandeis. The program got larger. And that was challenges with that. You know, we had Drosophila people who traditionally had not worked together. Mike Young, Jeff Hall, Michael Rosbash. That was being a referee among some tough folks. And so I was able to work my way through that, learn some valuable skills. And in the end, the sum was greater than the parts. I mean, they actually did great work together. So I think it was a fun time. And for me, it was just being around such smart people was a gift. Well, and I think there was some sacrifice. I mean, certainly in my lab, I kept it a modest size because I was doing other things. Remember, two things were going on. I was a center director, but in 1994, I think, I became vice provost for research. So I took on a major administrative responsibility at the university early on, too. So I was really doing dual timing, and that forced me to keep a lab that was a lot smaller than I would have. I probably had 3 postdocs most of that time, something like that, a few graduate students, 3 postdocs. And it was a struggle, but the work we were doing with the marine mollusk bulla was working beautifully. I mean, we had one good study after another. It was remarkable, partly because I had talented students. You know, Doug McMahon was a graduate student. He was doing wonderful work in the laboratory. I had Stefan Michel, who was doing very, very sophisticated work, electrophysiology. Good people all the way along. Martin Ralph was in the lab, while Chris Caldwell was in the lab, Nancy Wayne. It was really a remarkable stream of people who were productive. We were lucky. I mean, it had large neurons. You could study it. You could study it pretty well. I mean, it came together very nicely, and there were lots of good questions. So it was good. It was a good time. And you know, I was time sharing between the lab, the center, which really was quite different than my lab. My lab was really focused on electrophysiology. The center was focused more on molecular issues. So it was quite a bit of work at the time. And then I was, of course, trying to lead the research programs at the university. So that was challenging. And then in 1990, I have to think about dates a little bit, 2001, just at the end of the center's time, I became provost. So that was good timing because the center was then really finishing up. And I was able to devote more time to administrative work. But the center had a good run. I was amazed every year by the number of Science, Nature, Cell papers. But it was, you know, Steve Kay and his students and Joe Takahashi and his students. And it was a remarkable group. And when we came together, it was fun. I mean, I think it was fun being there together, talking science, and great times. It was unbelievable, and there were people behind the scenes that put in an enormous amount of work. I don't know if you know David Hudson, but Dave Hudson was one of the people. You know, Dave and I often, and this is something that's bound us together as friends forever. You know, we were 3 o'clock in the morning, we're going to Kinko's to get the grant copy to get submission the next day to drive it up to Washington. It was unbelievable, the people that put the hard work in, and it was several people who did it. It was an amazing amount of work, but we became a family working on it because it was all night. And in those days, as you know, with printers, you used to stand around all night waiting for it to get jammed. So you have to actually watch your work coming out very slowly from your Apple II computer. And it was an amazing time, and I think that we all put an incredible amount of work. But I still remember the tension on those site visits of getting ready for them. And NSF was very hard. I mean, they were back every year visiting, and they were very insecure about these centers because their great concern was these centers would not work. And many individual scientists had written to the National Science Foundation saying, this is a waste of money, that if you gave this to us as individuals, we could get much more science done. You're going to waste money with these centers. So the people were bitter about these centers because they thought it would take money away from individual efforts. Our center, in the end, one of the nicest things I heard at the end was the person who was the technical director of our center at National Science Foundation said this was one of the most productive centers they ever had. That in terms of the publications, in terms of moving the field forward, they were extremely satisfied, and I felt good about that. But it also had it's appropriate to have an end, and a lot of these centers that don't have an end start becoming less relevant as they go on. And other people have to have a chance. I mean, there was the problem that was developing in the U.S., maybe less visible here, the haves and the have-nots. And there was complaints that if you were in the center, you were getting the benefits of the center, you had the infrastructure, you had the equipment, you had all the opportunities. And if you weren't in the center, unless you were in a large lab, you really kind of excluded from a lot of these opportunities. So I think that the 11-year lifetime was perfect. You know, we accomplished what we needed to accomplish, and then others have a chance to build their program. So I think it was the right way to do it. I think NSF had the right idea when they had a sunset clause. But I've heard exactly the same comments from this because I've have enough colleagues in Europe hearing the people that are in, the people that are out. It's the same concerns.
Yep. They're saying we all wanted to be in that group, and you know
That's
we
right.
are in, and they are not. And,
Yeah.
It's the same thing, but you cannot imagine. Yes, you can.
Oh, yeah.
The amount of work that went into
Well,
to defining it.
right, what brought me back was, I think last year, Sergey invited me to Groningen when he was thinking about putting in a proposal, actually a Dutch proposal, and wanted to get my advice. And sitting around listening to everybody and listening to the guarded comments of how people were going to participate and what their concerns were, it just took me back. It was exactly the same position we were in at the time. I mean, investigators this is really a pretty insular business. People actually have, within your group, that's your family. But then everyone on the outside is sort of, those are the competitors. And it's hard to get into this idea of collectively doing science together. I mean, it's not natural for a lot of people.
It's not natural, and big science is, I don't think it's necessarily better, but linked
Right.
Networks
That's
that
right.
actually talk to one
Right.
another, I think are important.
Those can be much more productive. You can get a lot more done. And I just ran into that in the center, the advantage of having Steve Kay down the hallway to get advice when you're doing a project with such a different set of skills makes a huge difference. Mike and I are much more closer sort of intellectually, but we didn't, those kinds of relationships yield some types of advantages, but the people that are a little further away can make a huge difference. And I found, having people like Kay around and some of those folks in the center, for me, made an enormous difference in my research. So great advantages to it, but it's not natural. It takes a little while to develop,
The
center
territorial-
mentality.
Territorial
Yeah.
instincts.
The thing I learned about this, again, as I said, you can't underestimate the importance of your friends, of advisory boards. I mean, I just think the whole center, everyone in the center, after the first year site review, was really struggling. It really took the advisory board, some people not so emotionally involved, to help us think through where we needed to go. And it struck me as how collective sometimes the wisdom is that comes out of a large group of smart people, and that any one individual is having difficulty seeing how to go. We were really quite frightened, because we were convinced that our plan was a good one, our program, but we weren't obviously satisfying NSF. It took people from the outside to do that, and I thought it was remarkable how much time they spent, our advisory group, and I'm forever, thankful for what they did. But it wasn't us, it was really a combination of us and them working together, so quite impressive. Not being a beneficiary was easy, because they could make decisions, help me with resource allocation decisions, which were always emotional. How you handed out resources. The danger with centers, as you know, is that divide through by N, that everybody gets an equal share. And no matter whether they're productive or they're not productive, each year they get a guaranteed amount of money. And advisory boards are great at being able to give you some outside views on these things. So it was helpful, very, very helpful. So those were good years. And when we ended this center, I think we all felt pretty good about it. We also felt fairly tired, because the center was more than science. It was outreach, it was education, it was summer students, it was an enormous number of programs that NSF wanted in addition to science. So I don't know about you, Clark, but outreach, same issues.
Some schools, teaching,
Yeah.
public relations.
So Till's facing many of the same issues as he's directing this. Challenging. So I think we all felt it was a good time to end, but within 2 years, we, of course, were busy trying to apply for a new center. And, recently, we were able to get a Conti Neuroscience Center, which replaces in some ways the original NSF Center, but more focused, and certainly acting in a different way. And the field has changed so much. It's just one small center among so many efforts now. It's quite different. I think when the NSF Center was founded, it was large compared to what was going on around in the environment. Well, it was for those of us inside, as I said, the day that we found out we got funded, because it was never it was a long period of time when it was uncertain whether there'd be funding, and our university, I would say, was remarkable. And the university stepped up to the plate in dramatic ways, actually, to provide support along. NSF was looking for a large match. In other words, they wanted the institution to show that they loved us. And the institution was remarkable. It left me actually with a very positive feeling about administration, because our administrators were first rate. They decided what we needed, and they found the money to support us. And you don't always get that in an institution. It really was good. So it was exciting. And I think when we finished it, we were relieved, but we were excited about having been part of it. So I would say the remarkable thing about the field is it has not fractured yet. And so, there are a lot of fields that have made progress in the last 15 or 20 years. We're not the only field that has had remarkable progress. Many fields in biology have made progress, but many fields end up fracturing where you can't integrate from behavior, let's say, to the molecular activities. The groups begin to move into separate spheres, and they no longer actually talk to one another very effectively. They certainly don't have meetings together. At least they don't have small meetings together. And the thing I think that's been remarkable about this field is that, it started off with a Gordon conference that ran from behavior, as you know, in human studies to Drosophila and molecular studies. And I think when it first started, and this is very interesting, and I think it says something about our field. I remember the molecular biologists arriving for 1 or 2 days at the conference and deciding that that's all they wanted to stay for, because they certainly don't want to spend time listening to the people talking about human studies. Nothing could be learned there for them, or for even behavioral studies in animals, because really nothing could be learned there. And what happened over time is, unlike a lot of fields where people actually go off and create their own societies and their own interest groups, people stayed together and became more interested in what one another was doing. So now when you go to a Gordon conference, of course, everybody stays. And the molecular people, the same people that are now interested in the human work because they see the links and so effectively. So I think the integration of our field is remarkable, that people actually are deeply interested. People who are considered themselves chronobiologists come into this field from molecular expertise, endocrinological expertise, behavioral expertise, human work, and stay together as a unified field. So I think that's one of the great accomplishments. The amount of discoveries, of course, incredible. I mean, the discovery of clock genes placed the field above many other fields. I mean, memory suffered, for example, the study of memory from not actually having, sort of definable substrate and certainly didn't have mutants. So we had the advantages of great molecular biology, certainly some wonderful endocrinology and work on the brain, but it all has stayed together in a way that is so satisfying. I mean, it's integrated horizontally and vertically in ways that are really unusual, and therefore it makes for a great field, because it makes for a story. I mean, you can start off no matter what you're doing and talk about some very basic phenomena like jet lag, or more serious illnesses. And you can go from there to talk about basic underlying mechanisms, and that discussion goes on within a field that really has hung together very well. So I think that's where, if I have confidence in the field, the fact is we meet as a group, even though the group's very, very large now and very diverse in their expertise. I mean, I'm sitting and listening to sessions, and some of them you really struggle with. I mean, the specifics of the molecular biology is well beyond me. But you can glean enough from that you understand what the take-home message is, and in that same session you'll have discussions. In the same meeting you'll have discussions about, as you're talking about, you're dealing with depression, and you're dealing with that in the same meeting. That is remarkable. So I'm unbelievably optimistic. I think there's much to be discovered, and the field turns out to get more and more interesting because clock genes play other roles in control physiology and behaviors. So it's growing, not shrinking. It's not as if you're finding the smaller and smaller boxes you're opening up and you're getting to a point where people are wondering, is it still interesting? It actually is expanding in a lot of ways. So in one healthy field, it's changed. There aren't the great giants anymore. I mean, it's much more distributed in terms of the individuals. At one time when we entered the field, it was Pittendrigh, it was Aschoff. And that was good and bad. It was probably necessary at the time in the field to move the field along, to fashion it. It probably would be less useful today to have very strong personalities that were sort of dominating what they thought was important in the field. I think right now it's a mature field where I think probably the independence of having a lot of groups that all have quite a bit of authority, it could be very useful. And it's changed in dramatic ways. I mean, you can't look through the same leadership anymore. I mean, there are people like Mike Menaker, who are certainly senior leaders, or Sergey Don, but leaders in a different way, leaders who have never tried to actually strongly influence the way the field moves forward, and have just done their own good work and have had their own students, but have not played the same role, I think, that, for example, Pittendrigh and Aschoff played, which was essential in the early days of getting the field on the map and getting people converted, but maybe less useful when you have the maturity that we have today. So I'm optimistic.
That's lovely. That's lovely.